focused on modelling the weathering of petroleum products in the environment, and his master's research investigates the effects of data pretreatment on the multivariate statistical analysis of various forensic samples. He earned his bachelor's degrees in biochemistry, forensic science and application of science and law from Mercyhurst College in Erie, Pennsylvania, and he also worked as a police officer with the Ocean City Police Department before beginning graduate school. Please welcome John.
Thank you, Ruth. We've seen a lot of talks today that have shown a lot of different data analysis methods for forensic samples. So what I want to talk about is what to do with all of that data. Generally in a crime lab, a comparison is made between a questioned and a known sample. An analyst will then go into court giving his or her opinion on whether the two are consistent or not. But what happens if there's bias? What happens if a mistake is made? Those are some issues that were discussed in a 2009 National Academy of Sciences report. This report was put together by a committee that made recommendations on how to improve forensic analyses. There were a number of different recommendations that were made, 13, that focused on quality assurance, quality control standards, independent oversight of forensic laboratories. Much of the work that we've focused on at Michigan State has been looking at recommendation number 3, where we try to quantify reliability and accuracy in forensic analyses, look at the limits of accuracy and reliability in forensic analyses, and try to quantify uncertainty. So for an analytical chemist, that sort of translates to applying statistics. And so much of the work that we have done at Michigan State is trying to take widely available statistical techniques and apply them to forensically relevant data. We can break up the application of statistics in 2 different ways. The first is using a univariate statistical analysis, and that's used to compare individual samples or variables. So we can compare an average measurement using a t-test, or we can compare 2 different variances using an f-test. But oftentimes in analytical chemistry, forensic analysis, there's multiple variables. So then often multivariate statistical analysis are applied. These are used to compare samples and many samples and variables simultaneously. There's 2 different types that I'm going to be discussing. Unsupervised methods, where there's no prior knowledge of the relationship between the variables and samples, such as principal component analysis. And then supervised methods, where there is some prior knowledge and can actually be used to classify question samples. So in order to discuss these statistical techniques, I'm going to be talking about a couple of projects that are still ongoing at Michigan State. The first one is elemental analysis of paper, so looking at some elemental concentrations. I'm going to use this data set to discuss univariate and multivariate statistics, sort of compare and contrast them. This is a relatively simple data set where there are 5 variables and we compared about 80 different samples. Then I'm going to move on to a more complex data set, some chromatographic data, to compare the exploratory and classification techniques. The chromatographic data has, the way that we treat the chromatographic data is that each retention time in the chromatogram is its own variable. So each chromatogram has about, for our runtime, about 4,600 variables. We use that to compare 200 or so samples. And then last, I'm just going to touch on some of the challenges about implementing these statistical procedures into forensic laboratories. So as I mentioned, I'm going to be first doing some compare and contrasts between the univariate and multivariate statistical analysis on some document paper. So the objective of this research is to try to differentiate paper types based on an elemental composition. So this would be important in a forensic lab, generally a question document analysis. If there's like a will or something, there's a question of was a page inserted into a will? And so we can look at these different paper types and compare the elemental composition. In order to do this research, we collected 4 different paper types from the same manufacturer: color inkjet, laserjet, multipurpose, and office paper. We collected 2 reams of each, and from each ream used 2 sheets. From each sheet, we collected 5 different samples. Each sample was microwave digested, and then analyzed in triplicate by inductively coupled plasma optical emission spectroscopy, or ICP-OES. The result of this analysis is just an elemental concentration, or a concentration of each of the elements. We identified 5 elements of interest: aluminum, barium, iron, manganese, and magnesium, to look at. These elements were chosen based on their presence in all 4 paper types above the limit of detection and a low standard deviation between samples. So I'm going to show how univariate and multivariate statistics can be used to compare the elemental concentrations between the 4 paper types, but also within each of the paper types. So this is sort of the summary of the univariate results. We used a t-test to compare the elemental concentration of aluminum in each of the different paper types. Across the top and down the side are the 4 different paper types, color-coded, and also the ream A and ream B, the 2 reams, and then the 2 sheets labeled 1 and 2. The t-test was carried out at 95% confidence. If the samples were statistically different, it's highlighted in red. If they were not statistically different, it's highlighted in green. So we can see that within a paper type, there is no statistical difference in the elemental composition. This was true for all the paper types and was also true for all 5 elements. However, between paper types, there is a statistical difference in the elemental concentration. This wasn't necessarily true for all 5 elements, but as long as there is a statistical difference in one element, the papers can be differentiated based on this technique. The problem with these pairwise comparisons is that it's very time-consuming. To do these calculations, there are in this case, there are 136 different pairwise comparisons, and this was just for aluminum. So to do this for all 5 elements, there was 680 different pairwise comparisons. So in a forensic lab, that's not terribly efficient. For a grad student, I've got nothing but time, apparently. But this isn't going to be useful in a forensic, or it's at least going to be time-consuming in a forensic lab. So I want to talk about a method that's a little bit faster, is using the multivariate statistical analysis. And so specifically, I'm going to be talking about principal component analysis, or PCA. As I mentioned, it's an exploratory technique that can be used to examine the relationship between variables. There's 2 main outputs that we get from PCA, a scores plot and a loadings plot. The scores plot is a visual representation, or a scatter plot, essentially, of the samples. It can be used to associate and differentiate the samples. PCA maximizes the variance between samples. So the samples that are positioned farther apart on the scores plot are more chemically different, and then samples that are positioned close on the scores plot are more similar. The other output that we get from PCA is the loadings plot, and that helps us to identify the variables that contribute the most to the variance, or are the most important in differentiating the samples. The advantages of PCA: it helps in the simplification of data, and we've seen that at least alluded to in a couple of talks today. It reduces the dimensionality of the data set and helps to identify underlying patterns. So this is a scores plot of the different paper types. Ream A is shown as a diamond, Ream B is the squares for the 4 different paper types. The x-axis is the first principal component, accounting for 84% of the variance. The y-axis is the second principal component, accounting for 16% of the variance. So in this case, using only 2 principal components, we're able to account for nearly 100% of the variance, and that's because of the simplicity of this data set. We can see that there is the samples, the different paper types are discriminated from one another, and replicates and samples of the same paper type are clustered together. There's a couple that may be sort of an outlier, but the biggest differences are between the paper type, and this is supporting what we saw in the t-test and was done in a single step, a single calculation. We can also get the loadings plot, as I mentioned. The loadings plot, in this case, PC1 is the x-axis, PC2 is the y-axis. The farther away in this case, it's the variables that are plotted rather than the samples. And the farther away a variable is positioned from the origin, the more discriminating power it has, or the more important it is in discriminating the paper types. So aluminum and magnesium, which are positioned farther from the origin, are more important. Barium and manganese, which is positioned at the origin, really has no influence, or it really is not helping to discriminate the samples at all. We can use the loadings plot to explain the positioning of the samples on the scores plot. Office paper and multi-purpose paper are both positioned positively on the first principal component. Aluminum and magnesium are also positive on the first principal component. So that indicates that those two samples likely have a high concentration of magnesium and aluminum. And so when we look at the average element concentration in the 4 paper types for each of the different elements of interest, we can see that magnesium and aluminum do have the highest concentration that multi-purpose and office paper do have high concentrations of magnesium and aluminum. The laser jet paper, which is loading negatively on the scores plot, has a high concentration of iron. Iron is positioned negatively on the loadings plot, thus explaining the positioning of the laser jet paper negatively on the scores plot. So when we look at the average element concentration for the different paper types, we can see aluminum and magnesium are orders of magnitude higher than the other 3 elements. There's also a lot of variation between the 4 paper types in the concentration of aluminum. So that explains why magnesium and aluminum are important and are positioned farther away from the origin on the loadings plot. But we can't tell. So basically, these elements aren't contributing to the differentiation of the paper types. So we can apply a data pretreatment procedure, in this case scaling, it was logarithmic scaling, in which rather than plotting these on a linear axis, they're plotted on a logarithmic axis. This gives each of the variables a more equal weighting. So now we can see barium is actually varying quite a bit among the 4 paper types, especially because of its low concentration in the laser jet paper. We can perform the principal component after applying this data pretreatment procedure. And now we can see that barium is an important element in describing or differentiating the paper types. Actually, all 5 elements in this case are positioned away from the origin, so that all 5 are important in differentiating the papers, or can be used in differentiating the papers. We can also obtain a scores plot. In this case, the first principal component accounts for 94% of the variance. The second principal component accounts for 5%. So we're still accounting for nearly 100% of the variance. Now most of it's being accounted for in the first principal component because there are more elements that are being used to differentiate the paper types. Notice how far separated laser jet paper is from the other 3 paper types. Also notice the similarity of the office and multi-purpose paper on the first principal component. We can again use the loadings plot to explain this. The laser jet paper has a low concentration of aluminum, barium, and magnesium, and that high concentration of iron. So really all 4 of those elements are being used to differentiate the laser jet paper, making it positioning it negatively on the scores plot. The multi-purpose and office paper have similar concentrations of manganese, magnesium, iron, and barium. So that explains why they're positioned so similarly on PC1. The office paper has a slightly higher concentration of aluminum. Aluminum is positioned positively in PC2, thus explaining why office paper is slightly more positive on PC2 in the scores plot. So we can compare the loadings plots. Remember, the loadings plots are used to show the variables that are contributing the most to differentiating the paper types. Without applying the data pretreatment, there are really only 2, maybe a little bit of a third element that are contributing or helping to differentiate the papers. By applying the data pretreatment procedure, now all 5 elements are helping to differentiate those paper types. From the scores plot, we actually can still draw, actually in both scores plot, both with and without scaling, we can draw the same conclusions that we saw or we obtained from the t-tests. All of the papers are all the different paper types are clustered together and differentiated from one another, or discriminated from one another. By applying the logarithmic scaling, we're able to include more elements to differentiate the papers, thus getting more differentiation between the paper types. So we've shown that both univariate and multivariate statistics can be used in differentiating paper. The univariate statistics provide a comparison with a statistical confidence, but is time-consuming. The multivariate statistics provide a comparison with many samples, many variables simultaneously. The problem is there's still another measurement or calculation that would need to be made in order to actually describe how much differentiation or how much discriminating power or discrimination there are between the paper types. Another advantage, though, of multivariate statistics is that it provides a visual assessment of the relationship of the samples, which may in the future be useful in demonstrating these to the jury or to a jury. Last, we've also shown that data pretreatment, in this case scaling, can be used to help enhance the discrimination ability. And again, because we applied scaling, it applied a more equal weighting to each of the variables. Next, I want to move on to some of the chromatographic data and compare the exploratory analysis and the classification. For all of the chromatographic data, or for the chromatographic data, we're looking at ignitable liquids and fire debris. So the objective of the research was to relate simulated fire debris back to a known reference liquid or to a known liquid using principal component analysis or SIMCA. Generally, in fire debris analysis, fire debris is related back to an ASTM class rather than an individual liquid. So we looked at liquids from 4 different ASTM classes: the alkane class, the gasoline class, isoparaffinic class, and petroleum distillate class. And I'll show some chromatograms of those in a moment. All of the samples were analyzed in triplicate by GCMS, and we did generate some simulated fire debris. Because this is still ongoing research, we actually only have the simulated fire debris for the isoparaffinic and petroleum distillate classes so far, but we're working to generate the rest of the fire debris. The alkane class, we use 3 different liquids from the alkane class: lamp oil, lamp fuel, and candle wax lifter. The alkane class is identified by the presence of normal alkanes. In the lamp oil and the lamp fuel, there's a similar distribution of normal alkanes. And then in the candle wax lifter, there's a slightly different distribution of the normal alkanes. The gasoline class, we use 3 different gasolines, and gasolines generally identified by the presence of toluene, C2, C3, and C4 alkylbenzenes. The isoparaffinic class, we only use 2 liquids from that class, paint thinner and upholstery protector. And the isoparaffinic class is identified by the presence of branched and substituted alkanes. And last is the petroleum distillate class, where we use diesel, kerosene, and fuel injector. The petroleum distillate class is characterized by the presence of basically a Gaussian distribution of normal alkanes, as well as the presence of aromatic compounds and some branched and cyclic alkanes. So this is the scores plot of the ignitable liquids that I just described. The first principal component is the x-axis. In this case, accounts for 39% of the variance. The second principal component is the y-axis and accounts for 21% of the variance. In this case, we're only accounting for about 60% of the variance between the 2 principal components. That's because of how many more variables are included in this data set. We can use this scores plot with the exploratory analysis like we did with the elemental analysis of paper. Replicate samples are positioned close or right on top of each other. Samples that are chemically similar, such as the gasolines, are positioned close together. Samples from a class that are more chemically diverse, such as the petroleum distillate class, are more spread apart on the scores plot. We can also generate a loadings plot. In this case, the loadings plot looks very different than the plot we've seen previously. In this case, the y-axis is the loadings on the first principal component, and then the x-axis is the retention time, or essentially the variable. The advantage of this is it creates this sort of chromatogram-looking loadings plot. So we can actually go back and identify what peak this is based on its retention time. So that helps us to identify what compounds are actually contributing to differentiating the samples. So just to give you some examples, the alkane compounds, we can see the normal alkanes are generally positioned negatively on the loadings plot. Some of the branched alkanes are positioned positively. And for the aromatics, most of the aromatic compounds or all of the aromatic compounds are positioned positively on the loadings plot. We can use the loadings plot again to describe the positioning of the samples on the scores plot. The gasoline samples, or in the gasoline sample, all of the compounds are loading positively, so that explains gasoline's position positive on the first principal component. All of the compounds in diesel are loading negatively, explaining diesel's negative positioning on the scores plot. We can also look at how similar different compounds are in a class. So we can look at the alkane class. Lamp oil and lamp fuel, which have the same normal alkanes present, are positioned close to each other on the scores plot. Notice they're still able to be differentiated, and that's because of the slight difference in concentration of the C12 and C14 peaks. Candle wax lifter, which has a different distribution of the normal alkanes, is positioned farther from the other two. So we've seen how we can use the scores plot as sort of the exploratory analysis, but I want to actually move on to the fire debris and how we can, what we can do with the scores plot for assessing the fire debris. So we generated some simulated fire debris in the lab. This is a chromatogram of paint thinner that had been spiked onto carpet and then burned. Since we know what the ignitable liquid is, in this case it's paint thinner, we can compare the chromatogram of paint thinner to the simulated fire debris. And this is what's done by arson analysts. Generally, a comparison is made between neat liquids and the fire debris. Sometimes a total ion chromatogram is used, but more often they use extracted ion chromatograms or extracted ion profiles. Extracted ion profiles are just summed up extracted ion chromatograms for specific classes of compounds. So alkane class would use mass to charge, extracted ion chromatograms for mass to charge 43, 57, and so on. But just like the arson analysts, we can compare the peaks in the paint thinner to those in the simulated fire debris. We can use the mass spectral data to identify and verify that these peaks are the same. We can also, since we know that the carpet was burned matrix, we can see the compounds in the burned carpet that are also present in the fire debris. Again, we can use the mass spectral data to verify that. So essentially, this simulated fire debris is just a combination of the ignitable liquid and the matrix. However, what makes this analysis complicated is often the matrix and the ignitable liquid are unknown. So in a fire scene, often there's multiple matrices that are present. So it can actually make for very complex chromatograms. So what I wanted to do, or one of the steps that we took, was to try to use this scores plot as a model to predict or calculate the score or the location of the burned sample. So this would provide a visual assessment of sort of the relationship of this fire debris. So we can use the loadings plot that was calculated from this principal component analysis to project the fire debris. The advantage of this is since the principal component analysis is performed only on the neat liquids, that matrix contribution that we saw is basically going to be ignored. So we can project the fire debris, which is shown as the triangles, onto the scores plot. And you can see here it's positioned closest to the paint thinner. So this is a sort of a visual relationship we can, in order to assess the fire debris. It's visually, it's closest to the paint thinner. But we're not able to classify it. So in order to move on to classifying it, we wanted to use SIMCA. SIMCA, or Soft Independent Modeling of Class Analogies, is a supervised classification method. So what that means is known compound or known samples are assigned to a class. Principal component analysis is then performed on each class. Question samples are then projected onto the principal component model for each of those classes. So just like I did for the neat liquids, that's done for each of the different classes in SIMCA. And then a distance measurement is used to determine class membership. SIMCA is a soft classification technique, so that means that the results can be, a sample can be classified to sample can be classified to one class, no classes, or multiple classes. But the outcome from SIMCA is basically just yes or no, is it classified at a given confidence? So we had to develop a training set, or our set of knowns. Because we only had fire debris from 2 of the ASTM classes, we've only included in the SIMCA model these 2 classes. So that we assigned an isoparaffinic class and a petroleum distillate class. In each of those classes, there was a neat standard, so the paint thinner and upholstery protector, as well as simulated fire debris, which was the neat liquid spiked onto a carpet and then burned. The same is true for the petroleum distillates. We use diesel, kerosene, and fuel injector, both neat and as fire debris. We then also had a test set in order to test the model that consisted of the neat liquids, the simulated fire debris, and the burned carpet. So these are the PCA models for each of the training sets, isoparaffinic and petroleum distillate. As I mentioned, the training set, or the neat samples, are shown as the circles, the fire debris are shown as the triangles. As I mentioned, the test set is then projected into each of these PCA models. So the test set is outlined in red, so the neats are the squares and the fire debris are the diamonds, and we also put the burned carpet on as the black triangles. On the isoparaffinic model, the isoparaffinic compounds from the test set are positioned close to the corresponding liquids from the training set. In this same isoparaffinic model, the petroleum distillate compounds from the test set are positioned essentially at the origin, so they're not really being influenced at all in this model. For the petroleum distillate class, for the petroleum distillate model, the petroleum distillate test set are positioned close to the corresponding liquid, or the corresponding samples in the from the training set. In this case, though, the isoparaffinic compounds on the petroleum distillate model are actually positioned very close to the fire debris. I'm only showing the first 2 principal components. In this case, we used 4 principal components to develop the SIMCA model. So this is the results, summarizing the results of SIMCA. I have the different compounds listed, the total number of samples that were tested, the number that were classified as isoparaffinic, and the number that were classified as petroleum distillate. For the neat paint thinner and the neat upholstery protector, they were all correctly classified as isoparaffinic. For the neat diesel, kerosene, and fuel injector, they were all also correctly classified as petroleum distillate. All of these classifications were done at the 99% confidence. When moving on to the fire debris, the paint thinner and upholstery protector fire debris were correctly classified as isoparaffinic. And the diesel, kerosene, and fuel injector fire debris were correctly classified as petroleum distillate. The burned carpet samples were not classified as into either class. This is an advantage of the soft classification technique. If there isn't an ignitable liquid present, like there wasn't in this in the burned carpet, then it's not going to be classified to either or to any of the classes, ideally. I do just want to mention, though, this is a very simplistic and proof of concept model. Generally, in a forensic lab, we're not going to have known burned samples. And certainly they're not going to be burned with the precision of what we can create in the lab. Each sample was burned for the same amount of time, spiked with the same amount of liquid. So we got fairly consistent results in burning. That's not going to be true at an arson scene. So we want to take this model further. We've shown that this works. We've shown that we can classify the samples. Now what we want to do is take it further and try to use other things in place of the simulated fire debris. So a couple suggestions would be to use the extracted ion chromatogram, or some extracted ion chromatograms, instead of the total ion chromatogram to minimize the matrix interference. We're also thinking about starting to use evaporated samples to start to mimic some of the losses that are seen from the burning process. So this is, again, just sort of a proof of concept, but eventually we want to take it and develop the model that you can just plug in a sample from a fire scene and get back what class the ignitable liquid belongs to. So we've shown that we can use the PCA for an exploratory analysis that's fast, easy, good for a large number of samples. And we can also use the PCA to visually associate a question sample back to the neat liquid. And we've shown that we can use SIMCA to classify a question sample with a statistical confidence. Oops. So last, I just want to briefly touch on some of the challenges that remain in trying to implement these into forensic laboratories. The research is being done using multivariate statistics. We can see in the number of publications over the last 15 years, both for general scientific journals as well as specifically to forensic science journals, the research is being done referencing multivariate statistics. But it's not making it into the labs, and there's a number of challenges I think that are still that need to be addressed. We've seen in the talks today that there are a lot of diverse data types that are used in forensic analysis. Each different data type or evidence type is going to require research to determine what kind of model should be developed, what kind of statistical procedures should be used in order to classify in order to make the best use, I guess, of the statistical procedures. So in all cases, in the case of arson, we're trying to classify the samples. In drug analysis, the objectives might be different. With IR data, again, we may want to use some different statistical techniques. I didn't really talk much about the data pretreatment procedures, but oftentimes in multivariate statistics, data pretreatment is applied prior to the statistical analysis. The reason for that is often the greatest source of variation sometimes are actually coming from instrumental differences. So changes in retention time, variation in sample injection, things like that. So each of the different diverse data types would require a different data pretreatment procedure. Also, any statistical analysis is going to benefit from replicates, but has been mentioned a couple times. There are case backlogs in forensic science. So asking forensic laboratories to perform replicate analysis could prove to be problematic because it would create more case backlogs. Sort of along with that is to implement these statistical procedures into forensic laboratories. Analysts are going to need to be trained. Software is going to need to be purchased. This is going to require resources from the forensic lab that's already scarce. Time and money are in short supply in forensic labs. So to sink a lot of money into training the analysts and implementing these could be problematic. Also, if these are implemented into forensic labs, time's going to need to be taken at each analysis to perform and interpret the results. So again, that could lead to more case backlogs. And last, there's still some issues in the court system. There's still a question of objectivity. One of the things, in order to reduce human bias, one of the things that was outlined in the NAS report, we want to reduce the bias, so we want to make these procedures objective. But it's still an analyst that has to choose what techniques are applied, what pretreatments are applied. So there's still some subjectivity that goes into these statistical analysis. There's also a question of explaining these to a jury. Multivariate statistics are not the easiest thing to understand, and if a juror doesn't understand it, it's possible that they would simply ignore that piece of evidence, which could be devastating to a forensic case. And then last, there's sort of this question of acceptance. In order to get admitted into a court system, it needs to hold up to a Daubert or Frye hearing. If it's not able to, if these procedures won't hold up to those standards, it's hard to justify spending the time, money, resources in order to implement them. But it's hard to it's hard to realize what the full potential of these statistical procedures are until the research is done and until they're implemented in the crime labs and get into the court system. So in conclusion, shown that the multivariate statistical procedures allow for comparison of samples and many samples and variables simultaneously. They're rapid and suited for large data sets. And these procedures can provide a statistical assessment of reliability and accuracy in forensic analyses, addressing some of the issues that were named in the National Academy of Sciences report. Just last, I'd like to acknowledge our funding, the National Institute of Justice and the NIJ Forensic Science Foundation Student Research Grant, Analytical Sciences and Dr. Kathy Severin for instrumentation help, Emily Riddle, Andrew DeJarnett, and Jordan Geiger. They were the graduate students that collected all of the data, and the Michigan State University Forensic Science Program. And with that, I'd be happy to take any questions. Yes.
This might be a fun question, but I was wondering about the software. Do you have any recommendation for reflecting data treatment?
Each software has their own advantage and disadvantage. We're currently using 3 different ones in the lab. MATLAB with the statistics toolbar, Pirouette from Infometrics, and the Unscrambler from Cameo. They all have certain advantages and disadvantages, and I'd be happy to sort of talk more with you individually afterwards and discuss if you have something specific that you're looking for, maybe guide you in that direction. Yes?
Have you looked ahead to any other applications? You talked about fire arson. Have you looked into any others that the statistics might be more helpful with?
In our lab, we've done gunshot residue. I've done a little bit of work with determining time since death, looking at volatile organic compounds. Controlled substance analysis is a huge one. We do profiling of ecstasy tablets, and so identifying impurities that are indicative of maybe a batch where an ecstasy tablet may have been created. So there are a lot of different areas that at least our group has looked at. In the literature, I would hazard to say that any forensic evidence probably has it applied somewhere. I think that's probably a safe bet. Yes?
I'm just curious, on your arson stuff, what if your personal interest is a mixture? All of you use a mixture.
I'm sorry, can you say?
Your personal interest on the arson work, what if you use the mixture?
We've actually started. So what if we use the mixture in of ignitable liquids or something? Yeah, we've actually started looking into that a little bit. I think probably principal component analysis would be a starting point, but there may be some other statistical techniques, PLS or something like that, that may help more in differentiating them. I'm not sure, but we've done a little bit of research looking into that. Any other questions? Thank you.
I'd just like to thank our speakers once again. And thank you all very much for attending this afternoon.